It is my pleasure to introduce our next speaker, who is Augusto Rendon from Genomics England, from the UK, obviously. And Augusto Rendon has been with Genomics England for almost 10 years now. He is the chief bioinformatician and oversees a team of more than 70 bioinformaticians and data scientists there, also involved in large-scale genomic projects. And he will take us to the UK and let us know about how to make the most of genomic data for clinical and research. And he will take us to Genomics England's clinical and research infrastructure for genomic medicine.
Thank you so much for the introduction, and thanks for the invitation, and congratulations on all the good work that GHGA has been doing. I decided to cut the research bit of the talk, so I ended at health. And the reason is I think we will hear lots about the research. Is something that we present often, and it's something that is easy to summarize. We talk a lot less about health, and given future programs here in Germany, it might be good to show a little bit more onto how we do the healthcare part. I hope that's okay. Good. So just to give you a feeling for Genomics England and what we have been up to, we have this timeline. First thing to say is Genomics England is what's called an arm's-length body that is part of the Department of Health and Social Care. All our money comes from the research arm of the Department of Health. The other arm would be the NHS that pays for health. We were originally created in 2013 to launch the delivery of the 100,000 Genomes project in the UK. And we finished doing that between end of 2018. Results were still flowing through 2019, but all the sequencing was done then. During the pandemic, it was great that the government asked us, could you please sequence some genomes from individuals with either severe or mild COVID. And so we ended up collecting about 30,000 of those that are available for research. At that same time, and I'll talk a little bit about this, the UK was launching a genomic UK strategy that tries to say, what are we trying to accomplish as a country? And importantly, and what I'm going to talk most about is at the end of 2020, we launched the NHS Genomic Medicine Service, which is the provision of a reorganization of the way genetics is delivered in the NHS and oncology. And I'll show you lots more about it. Since then, we have been busy by further funding on things like newborn sequencing, a program called Generation Study, which is already ongoing. We haven't started recruiting. We expect to do that at the end of this year, 100,000 babies, newborns. And the idea there is to do screening in parallel to the standard pill prick tests to see if whole genomes can improve the screening situation. We've also done a few other programs or are in the process of doing large long-read sequencing and also increasing the multimodality of our data, largely around imaging and collecting more information and understanding better how what we do can benefit from diverse data populations. So sequencing, primarily at the moment, we're sequencing populations with sickle cell anemia that tend to be underrepresented from both an ancestry and from a healthcare perspective, or underlooked at. So going back to that Genome UK strategy, it has 3 pillars. The first pillar is research, having data sets that enable us to do research. We are not as lucky as the Danish with this wonderfully coordinated biobanks. We have a number of them, some of them large, like the Biobank, UK Biobank. NIHR Bioresource is less known out there, but it is about 200,000 people that have been all genotyped, and they can be recalled based on genotype for further studies. And there's a lot of that going on. The second one is prevention. How can we learn about intervening earlier? And the main study there is Our Future Health. They have recruited about, I think, 800,000 people now. The idea is 5 million. They're all SNP typed. and they have longitudinal health records that come from secondary sources with the idea of, for example, developing ever better predictive scores and so on. And lastly, we have, that didn't go well. We have a diagnosis and personalized medicine where indeed Genomics England, in collaboration with the health service, have been, or the other way around, have been delivering, essentially implementing some of these technologies into healthcare. And that's what I'll talk about only. So how we do this at Genomics England, we tend to look at the world through this infinity loop, where on the left-hand side, we have efforts to evolve genomic healthcare, and on the right-hand side, research. And so if I take you through this, there is a doctor that saw a patient, there's a test that gets ordered, all of this is happening within the NHS. Then we handle the sequencing, analyses, and return those results back to clinical labs that, or analyses back to clinical labs that interpret them, return back to the patient. Subject to consent, then those data sets, both the test ordering information that includes phenotypes, types of samples, and so on, are placed into a research environment, a trusted research environment where, in this case, it's the identified researchers, pharmaceutical companies, and biotech, and so on, can come and do discoveries. As discoveries are accumulated and with efforts from ourselves to increase diagnostic rates, which I'll talk about, those data flow back to the patients, and that's the idea of this infinite loop. Cool. So as I said, I will focus the majority of what I'll say on this side with a little bit of diagnostic discovery. So I mentioned we had delivered the 100,000 genomes project, and it was successful, at least in sequencing 100,000 people, or sorry, 100,000 genomes. We did that both in patients with rare diseases and their families. There were roughly 35,000 families, 70,000 or so people with members of a family with rare disease. We did thereabouts of 17,000 tumor normal pairs, so patients with tumors and their corresponding normal sample. Somehow that adds to over 100,000. And we returned diagnosis from that. And as a response to that, the NHS decided to start funding whole genome sequencing for standard of care. And they also reorganized the way that genomic laboratories were operating. It came from over 20 laboratories across England. Health in the UK is devolved, so each nation has their own budget. So this is only England, as you can see. And what that new genomic medicine service consists of is primarily two things. The first one is the amalgamation of those laboratories into 7 regions and 7 laboratory hubs, as we call them, if I use the term GLH. They are distributed to provide care across 55 million people. And one of our big things is to be equitable, so that if you're seen somewhere up north, you get the same sort of quality that you'll get, say, in Exeter. So that's very ingrained in our NHS constitution. A big thing is also operating to common national standards, specifications, and protocols. And finally, a single consent that enables patients to then participate in research in that infinity loop flowing into the right-hand side. The other big part of this, and there's all the pieces, but the other big part is the formalizing of the directory where for each condition, the types of genomic tests that a clinician can order are stipulated and that will be paid for. So I'm giving you here the rare diseases part of the test directorate, but there's also a cancer part to that. It's all publicly available if you want. And if you can see here, perhaps it's too small, but a subset of that is whole genome sequencing. For a number of indications, I'll go that in a second, you can order a whole genome sequencing test from any hospital in England. Currently, while the number of indications just under 40, maybe 5% of tests that are genomic tests are whole genome sequencing tests. There will be many panel tests and MLPAs and FISH and things like that. Cool. So if we look at in more detail, what we're doing in rare diseases is precisely the types of things where we had seen in the 100,000 genomes, high diagnostic rates, and where you're able with a single test to be able to replace other existing modalities. So for example, some of the indications appeared when we were able to confidently call short tandem repeats out of your whole genome sequence, because that meant that a set of additional PCRs or whatever didn't have to be done. And that's been the way we're evolving this, adding more functionality to the pipelines, and then identifying populations of patients that can benefit from whole genome sequencing. And then we also have cancer indications. These are not the large solid tumor populations. They are those where you either have very good evidence that there are things like structural variants that are determining the treatment of those, such as in leukemias and sarcomas, or where it really makes a difference to spend a lot more time getting a fuller test, such as in pediatrics, relapse and refractory. Since 2020, October, when we launched, as of maybe a month ago, we have sequenced in rare diseases 28,000 families and about 3,000 patients. Cancer is going quite slow. Rare diseases has stabilized at around 4,000 genomes per month. Same issues that Soren was mentioning. Clinicians are just not ordering as much as they could. So how we operate, we have, because of the legacy of the 100,000 genomes, how we were integrated to try to push the health service to adopt some of these technologies. Genomics England works in a, I would say, high friction with the health services situation, where the information that is traveling between the health service and say Genomics England is quite rich. And I'll say a little bit more about that. Let me just take you through the circle. So as I was mentioning, patients see doctors, the sample, there's a test ordering event where it says, I want this type of test, here's my pedigree, or here are my specifications of tumors and normals and so on. There's consent taking, and so on. There's then DNA extraction. Then there is a bunch of communications around plating those DNAs and sending us to Illumina. Illumina laboratory services in Kingston, Cambridge, are subcontracted from us to do the whole genome sequencing. So we only have, in this case, 4 whole genome sequencing one site. Then they send us the data. We do all the alignment, variant calling, and we run a number of prioritization and classification algorithms. So we don't return a report. Rather, we return a set of analysis back to one of these GLHs to review the data and interpret and say, I think say in the rare diseases scenario, the 2 variants that together are explaining this condition are such and such. We don't do that. They remain liable for that as well. So then they feed that report onwards to the patient. We do that, all of us, on their in vitro device diagnostic certification and medical laboratory ISO 15189, a few other standards. And the reason I was saying that the interfaces are quite big is if you compare this with, say, a blood test where you maybe order something very simple in a EPR and then send some bloods, and all of that, all the way to a result ready for the doctor, happens in a single loop. We have to exchange quite a lot of data. We're exchanging a full annotated set of genomes here, quite a lot of sample tracking, kind of LIMS stuff here. The reason I'm emphasizing on this is definitely we don't see this as the future. So one of the main views of this GMS is that by bringing all those laboratory hubs together, you can then operate as a national virtual laboratory. So you no longer have your little database of variants that you might have seen before, or the 100 genomes that you can use to develop new things. You really want to operate as a single virtual laboratory. And we have been doing that. And I will show you next some of the things that we have doing in that space. The thing that I'm not showing you is the test ordering system side of things, how you can do the consenting, test ordering, in large part because we are really trying to move that back to the hospital record systems, away from having a portal where the clinician has to remember how to log in, although now that's the same login. They have to remember how to phenotype a patient. We want to really remove that boundary there, that friction, by ordering directly from the EPR. So it's a bit of a dream, but that's hopefully in the next 3 to 5 years that will happen. So the main areas where we have put effort in enabling that virtual laboratory have been in creating API exchange to exchange data. And so today, across all these genomic laboratory hubs, they can read genomic data through HDS GET as a service from each other. If you have the right authentication, they can export and read and return as well genomic analysis, the idea of like a prioritized classified genomes. Also the reports and their APIs that are well established that are available open to those genomic laboratory hubs as well. Next, these are, of course, essentially JSON documents of sorts. We have also built and allow and manage the integration of third-party tools to receive those results and interpret those genomes. So we call them decision support tools. These are the types of I'll show you an example in a second. So we have been building and integrating a variety of them, working with third-party vendors, and as I said, with them. Importantly, we have been developing knowledge bases. And so if you think about this central project, it's the same as in the Danish project, one of the things we can offer nationally is the careful curation of data centrally so that everyone can benefit from that. I'll give you a few examples, but that includes things, particularly things like how do we manage gene panels as a country? How do we manage case level information? And less relevant, perhaps because it's more static, is genomic annotations. You know, where are the amino acids, but also tell me the latest splicing prediction score. One thing that we are not doing nationally, but we are definitely re-platforming after 9-10 years of experience, is how we do workflow orchestration. And we are doing that now in a hybrid way. We have both compute and storage on-prem and on the cloud and Amazon. And we have then completely adopted what are by now quite commoditized things such as Nextflow Tower, and essentially hooked up little standard things such as event streams and so on to manage those workflows. We are starting to plan to collaborate with the GLHs about taking the workflows that we have developed so that we can then validate it so that they can use them as well. And finally, one of the things that we have found most difficult is, in the example that I was giving about short tandem repeats, getting those pipelines validated meant going for almost 2 years to the various labs across the UK to say, do you have some standard of care results that I also have genomes for? And then can I try algorithms to be able to say that I'm meeting standard of care? So you can imagine bringing from all the regions sufficient numbers to be able to validate this. We're talking about 200, 400 participants, the same for CMVs and so on and so forth. To be able to bring those, it's very time consuming and therefore slows down innovation. So we are proposing to bring that more centrally across the GLHs. Again, as I said, we are using and doing some of this internally. We're working with the NHS to do that as a country. So let me give you some examples. Here is just one of those decision supports. We are just launching this. It's, I think in October, we flipped from the system we were using to this system. Here's just one view, but it allows you to look at variants of different types. In this case, I'm showing you the structural and copy number variant views. And one of the reasons we decided to spend money in building these tools is that, particularly in cancer, the speed at which your pipelines, your analysis are changing is still quite high. It's not like a lot of you might be doing research to bring new biomarkers, new ways of analyzing data. And therefore, it makes a lot of sense that you want the interfaces by which you are delivering those insights to your users, in this case, to the laboratories, need to change and work hand in hand. In the past, we had like third-party systems, and the advances we had in the pipeline were just not making it through. So there's an area, particularly in cancer, where we decided to build. And so you can also get genomic signatures, and every time there's a new biomarker, I know, homologous recombination defects and detection of that, you can put it in there. In the rare disease situation, we're actually using more commoditized tools, just because there is less happening in that space. There aren't necessarily new types of ways of looking at the genome that are different. So interpretation. The second thing is panel up. It started very early on, but this is essentially a way of maintaining lists of genes. And we use that for each of those indications that are panels or whole genomes in the genomic test directorate, there will be an entry here. We've been working on that. And finally, this is what we call the clinical variant arc. This is what I was talking about. For each case that goes through, we store all the case information. So that would be some of the phenotypes that are presented in the patients, the family structure, the various members of the family, and essentially all variants that were classified or considered clinically relevant, be it from benign to pathogenic, be it by algorithms or by humans, as they are interpreting those. And that is connecting in real time with the interpretation systems. And this is real numbers. So we've had about 63,000 families in both the 100,000 genomes and PMS, and some numbers of variants, diagnostic rates, and so on. But the idea here is this is the sort of thing that the GLHs can then go and say, oh, I'm interpreting this person, this variant looks interesting. How many cases like this one? Who else has had this variant? Is this a variant that is propping up too often or not? Or what are variants that have been deemed pathogenic, and can we just prioritize them immediately in these types of samples? Those are all things that are happening. Let me finish then with just a little bit around the loop, which is that diagnostic discovery. So we are returning data to the GLHs. They're making discoveries. They're making diagnoses, but we recognize that they only have so much time to interpret the genome in a clinical service, and that they only have a case rather than having the luxury of looking across cohorts. So we have an ongoing effort, which we call diagnostic discovery, where they take a cohort view approach to identifying new bioinformatics, new diagnosis, and they change a little bit our bioinformatics pipeline so that they can do something a little bit different and add new annotations without having to validate things. And so just to give you an idea of why that is valuable and why I'm bringing it up is, on top of the diagnosis that had gone through the first round of the 100,000 genomes, they added 35% increase. So that is equivalent to more than one of those labs. The majority, 85% of those findings, when they return them back into the health service, end up being reported. And they continue to, it's not plateauing the number of diagnoses that they're providing. That's 2,500 families that have benefited from that effort. The types of variants that they're getting, this is not a systematic approach. So they essentially look at, oh, there's a new gene. Let's look at who else could be benefiting from that gene. Or, oh, now we have a structural variant calling that is better. And so let's look at all the structural variants and those sorts of things. And it gives you an idea of where differences are coming from. And that's that. Let me just finish with some parting thoughts. We definitely started from a world where those big Illumina machines needed to be fed because they were huge monsters. And you had to sequence to be cost effective, tens of thousands of samples. That's not the case. And we're really gearing towards a future in 5 years where more of that is happening in a distributed way. And so where we want to focus then, and where we're gearing up to focus, is collaborating on workflows, on compute and data infrastructure, so that we can all share the difficult, the difficult bit is getting up in the infrastructure, making sure you manage the data. That's what you can centrally develop, and where you can have expertise. Then I said that just a second ago, doing the heavy lifting. The idea that bringing more data together definitely helps patient outcomes. And so why do we share data? We do that because ultimately patients will do better. And just to finish, NHS England is doing quite a lot of effort and putting money into creating something called a unified genomic record, which my Danish colleagues will say, wow, we've been doing that for a while, but we're just getting there, which is the idea that every single genomic test that I spoke about then gets a record that you can then go and revisit in later time, both for clinical delivery. So what was the report? Where were the files? What was the analysis? And also from a research perspective, being able to link all those panels, all those exomes, all those genomes into the same place. That's all what I have to say. I leave you with some, thank you very much, and some links if you want, if that's very helpful. Thank you.